Yes, so this phase one trial, I think this is very um, is something that's now done by a lot of biotechs and pharma, that you also use the phase one trial to get a little glimpse into the therapeutic window in a in a disease of interest for a drug.
Hello, you're watching Proactive. I'm joined by Immunic Chief Medical Officer, Dr. Andreas Müller. Andreas, good to speak with you. Data from your phase 1/1b clinical trial of IMU-856 published in the peer-reviewed journal, The Lancet Gastroenterology and Hepatology. So quite an accolade for Immunic.
Stephen, it's a great opportunity to talk to you today because I think we're very proud and very thrilled about the publication of our data in Lancet Gastroenterology and Hepatology. It's a phenomenally good paper and publication with a high impact factor, and this also speaks to the importance of these data for the medical community, that they're so interested and basically have it in this very prestigious paper. So we wanted to thank all of the authors, the investigators, the patients who have participated, that really enabled us to get to that stage that we could publish now the full data. And we are hopeful that this also leads to more public awareness of this drug, IMU-856. This is a drug that is in clinical development, and this is a clinical candidate from Immunic, our second clinical candidate. And it's, to my mind, and in my personal view, it really is very different from other approaches in celiac disease, but also in other gastrointestinal diseases. So I'm very excited about this drug. I've always been excited, and hopefully this leads to allowing us to really quickly go into further clinical studies and also exploring this in other indications other than celiac disease.
Could you give us a summary of the phase 1 data in healthy volunteers, Andreas?
Yes, so the publication covers the complete phase one study that we did in Australia and New Zealand. And it basically falls into three parts. One is healthy subjects are used in part A and B, the first part. So this is where you increase doses in single and multiple doses initially, and you do this step by step in order to explore the safety and the pharmacokinetics, so how the drug distributes and is excreted in the body, and learn a little bit more in terms of how you can dose the drug and what the safety and tolerability is. And we have seen in this parts A and B in healthy volunteers that the drug is very well tolerated. There was no systemic or general increase of any adverse event, any lab abnormality, anything that seemed to be related to increasing doses of drugs. So that was the good news. We also found that the pharmacokinetics, so the giving the drug as an oral treatment, leads to a very good uptake and distribution in the body in a way is around with a half-life about 20 hours. It means it confirmed that this drug can be given very conveniently as a basically single oral administration daily, which is also good news. I think we were quite pleased with the overall features of this drug that we found in humans in this part A and B. Yes.
So how about the phase 1b data in celiac disease patients, Andreas?
Yes, so this phase one trial, I think this is very is something that's now done by a lot of biotechs and pharma, that you also use the phase one trial to get a little glimpse into the therapeutic window in a disease of interest for a drug. And we did this in what we call part C, which is also published in this paper, where we targeted patients with celiac disease. And celiac disease is, from many angles, a very interesting patient population. I think, first of all, it's an underserved patient population. There's no treatment. It's a serious autoimmune disease with a lot of complication for these patients, a lot of problems in quality of life for these patients. And there needs to be treatments. But also, I think for us, it enabled us to do a proof of concept that the drug potentially could work in different gastrointestinal diseases, because celiac disease is a disease that allows us to do what's called gluten challenge. Gluten is a part of the autoantigen, and so you can give a gluten challenge and can measure the effectiveness of your drug to prevent gluten-related increase in symptoms and in deterioration for these patients relatively quickly, because in a phase 1 trial, we were limited to a 28-day dosing for this drug at the moment. At that time, of course, we have additional data now, but I think this really enabled us to have a clinical proof of concept in a very relevant indication of gastrointestinal disease with a high medical need.
Andreas, can you provide a quick summary of the mode of action of IMU-856?
Yes, so this, like I said, I like the mechanism of action because it is very different from other approaches, because a lot of the autoimmune diseases or chronic inflammatory diseases are treated with immunosuppressive, immunomodulatory drugs, which has a lot of disadvantages. It helps to ameliorate symptoms and the disease itself, disease activity, but it also has long-term consequences, for example, for infections for these patients. So this mechanism of action really spoke to me when we first saw this drug, and it spoke to me because it is targeting the epithelial regeneration in the bowel, meaning it triggers, and that's what we have seen preclinical, also what we've seen in this phase 1 trial. It triggers the physiologic process of renewing the epithelium in the bowel without having a direct effect on the immune system. So it basically allows this regeneration without having the long-term side effects, we believe, of these immunosuppressive, immunomodulatory drugs. And as a secondary effect, more or less, you also have an effect of the barrier function of the bowel. Yes, because the gut or the bowel is full of bacteria. It's called microbiome, and the bowel has the function to prevent these bacteria to have any effect on the immune system, which is in the bowel wall. So the bowel barrier function is very important for many diseases, and it has been very well, I think, established now over the last 1 or 2 decades. And so we've seen also in preclinical experiments this the strengthening of the bowel barrier function. And actually in the clinical data, what we had seen that it regrows the epithelial villi, the architecture in the bowel, or prevents the damage of gluten on these architecture. It also shows that we strengthen the bowel barrier wall, and we saw this with some of the biomarker data that we have. We also prevented or ameliorated symptoms of gluten-related exposure. And we also, that is really, I think, very, I think a great outcome of this clinical this phase 1b trial is that we enhanced the uptake of micronutrients and important vitamins, for example, for these patients, because that has never been shown, to my knowledge, in clinical trials for celiac disease. And most of the complications celiac disease comes from the absence of certain, or the less uptake of these micronutrients or vitamins that are essential for us, and that has also been shown. And this altogether, I think, makes it a very interesting and appealing mechanism of action for celiac disease, but also for other gastrointestinal diseases.
I mean, what's the potential you're seeing for other gastrointestinal diseases?
So this is this bowel barrier function and the epithelial architecture is of so much importance for many gastrointestinal diseases. And this goes way beyond celiac disease. And there's lots of ideas why this should be applied to other gastrointestinal diseases as well. So this is inflammatory bowel disease like ulcerative colitis, and Crohn's disease, of course, is kind of that comes to your mind right away. There could be also use, for example, in what's called diarrhea predominant IBS, which is kind of a symptomatic disease for many patients because it's also driven by bowel barrier function. There is other diseases where this could be applied to. So we have to think about it a little bit where we go beyond celiac disease. Celiac disease definitely is something that we like as an indication and we would like to go forward. But we also think we should expand this to other gastrointestinal diseases to really see the full power of this mechanism of action. And hopefully this publication of our phase 1 data in Lancet hopefully helps to achieve that.
Congratulations again on the publication. Thank you very much for speaking with us today.
Thank you, Stephen.
As Immunic Chief Medical Officer, Dr. Andreas Müller.